And we have our last speaker here from the Walton Center.
My name is Amr. I'm a neurosurgeon at Walton Center and also second year MD student at Hull University. I will talk about the cytokine release in glioblastoma and being live in microfluidic chip. As we know, the key challenge in glioblastoma is the complex microenvironment that GBM have. And unfortunately, a traditional method like animal models or 2D cell cultures fell short to replicate the complexity of its microenvironment. So no better option than the patient GBM tissue that can mimicking closely to the microenvironment of GBM that allow me to testing the therapeutic interventions. So in the next 4 minutes, I'll show you the steps that we did developing the microfluidic chip that maintaining GBM tissue from the patient for 8 days and in some cases 12 days in the lab. So the usually step is during the surgery, ethically consented patient, we take a part of GBM tissue that doesn't need to send it for histopathological diagnosis, and we put it in the nutrient media DMEM immediately and transfer it to our lab within 1 hour. In the lab, we sectioning the GBM tissue in small pieces and put it in our microfluidic cylindrical shape, as we can see, with the inner tube connected in one side to the syringe with the nutrient media in control and also with or without treatment. At the treatment, we tested the temolizumab, the classic one, plus the protein arginine methylation inhibitors. On the other side, it's connected to a effluent tube collecting the cytokines released. We put that in the Harvard syringe pump with a rate of 3 microliters per minute with the nutrient media over the 8 and 12 days in the lab. We're collecting every 24 hours the effluent tube, connecting the cytokines. We store that in freezer minus 80 to maintain the cytokine integrity. We used, to test the cytokine, we used the protein profiler membrane from RDN Systems. It's allowing 105 human cytokine tested in single membrane in duplicate form that assuring the accuracy. And we also making sure that we double check and validate part of this 105 cytokines by ELISA. There's a huge amount of data over the 12 days of the 13 patients that we already done, but for purpose of this presentation, I'll pick 2 interesting results. This is a real picture of the microfluidic setup. And the first one is, we comparing the tissue before we putting in the microfluidic chip, before and after until in 8 days and 12 days. And we can see from the H&E staining that the GBM tissue cells maintaining its integrity after 8 days on the chip and also in the 12 days maintaining its integrity. And that confirming the visibility and of relieving from GBM tissue in the microfluidic, maintaining its integrity and no much difference over the first 4 days. But after the 96 hours, which is the 4 days, starting to decrease and change patterns, and even more in the 12 days. Also, when we look to the interesting cytokines, the most abundant cytokines released by GBM tissue under treatment, we found that TGF and MMP9 are significantly reduced under the novel treatment that we tested in this case, comparing to control, and also found that chitinase-3 like 1, which is a cytokines related to the cancer colon mainly, but in the GBM we found that it's really abundant and increased against the treatment, which can be a rule of resistance. To conclude, I think the study illustrates that visibility of maintaining glioblastoma for 8 days and in some cases 12 days, while we are allowing testing against control the different treatment that can allowing this study and this model of microfluidic to be a potential GBM model to test therapeutic intervention and see how much it can affect over the 8 and 12 days. Of course, for more information and things, happy to receive any email. Thank you.